Hello friends, I am Dr. Prashant Sharma and you are watching MEDICO'S Hub. This is a series of lectures on complement system, and in the third video on complement system I am going to discuss the alternative pathway. So alternative pathway, again there will be 4 steps in which it will be discussed. First one is initiation of pathway, then formation of C3 convertase, then step 3 is formation of C5 convertase, and finally the formation of membrane attack complex. So this pathway is stimulated or initiated, that is alternative pathway is initiated when some complement proteins like C3 come in direct contact of certain antigens which may be present on the surface of microbes, etc. These antigens may be like IgA, IgD, nephritic factors, or most importantly these may be endotoxins. There may be cobra venom even. So these are the various antigens when complement proteins like C3 come in direct contact with certain antigens. For example, this is an antigen. So these antigens will result in directly spontaneous hydrolysis of complement factor C3. So it will be hydrolyzed to C3A and C3B. So this spontaneous hydrolysis of complement protein occurs when it comes in direct contact of certain antigens. Now, when C3B is formed, it will be attached to surface of pathogen. Then, in presence of another protein B, it becomes C3B, capital B. Then in presence of factor D, it undergoes spontaneous lysis. Which factor? B will undergo lysis to form BA and BB. B small a will be released and B capital B will remain there. That is C3B capital B small b, with release of capital B small a. This is unstable complex. It is C3 convertase. This complex is C3 convertase, but it is unstable complex. So in presence of properdin, which is also known as factor P, it becomes C3B capital B small b capital P complex. So this complex is known as stable C3 convertase. Now, when C3 convertase is formed, that is formation of C3 convertase has occurred, it will result in lysis of C3. Another molecule of C3 will be lysed and C3A will be released, while C3B will be formed, which will combine with C3B BBP complex to form C3B capital B small b, another 3B properdin complex. This complex is known as C5 convertase. This is C5 convertase. And as soon as the C5 convertase is formed, the rest story is same that was in case of classical pathway. C5 will be lysed to C5A, which is released, and C5B, and this C5B will recruit 6, 7, 8 and many molecules of C9 to form the membrane attack complex to C5B, 6, this is C5B, 6, 7, 8 and 9. This is known as membrane attack complex, and this will be formed in the membrane, resulting in the formation of holes or pores in the membrane, and the cell will die. So this is the alternative pathway, which starts with C3, capital B factor is involved, factor D, then properdin is involved to stabilize it, then C5 convertase is formed, and then membrane attack complex is formed. In the next video, we'll discuss the lectin pathway or mannose binding lectin pathway. Hit the like button, share and subscribe our channel to get the latest updates and notifications.